Okay, hi everyone. This is just, I'm going to try to make this short, little introduction to that mTOR paper. Also, I'm just going to review the last few slides of the cell signaling lecture where I talk about mTOR because obviously it's related to that paper. Okay, so what we were, if we kind of start from here, right? We were talking about tyrosine kinase receptors and how they signal, right? And basically, talking about growth factors and cytokines, right? And so I gave you some examples showing you some pathways of cytokines or some signaling pathways that are initiated by the binding of cytokines and also by the binding of growth factors, right? Okay, so growth factors, cytokines, and then there was this summary slide where we basically compared the effects of growth factors versus cytokines, right? And to see that they are basically opposites of one another. Now, mTOR, okay, which is what this paper focuses on, and really it's just this mTOR C means mTOR complex, right? A complex of proteins. And I believe most of them or all of them are kinases. I know some of them are kinases, which phosphorylate proteins, okay? So this is giving you an example of a different pathway, right? So if we go back here, this was showing you a certain growth factor pathway, right? Just giving you an example of one. And remember, we're not memorizing any of these, okay? This next one is just another example of a different type of signaling pathway that would be initiated by a different growth factor and a different receptor, right? Remember, we talked about, I think I mentioned different types of growth factors like PDGF, there's EGF, there's also like growth factors that are specific to the nervous system like NGF, BDNF. So there are a lot of different growth factors, okay? And so which one of them is going to signal differently, okay? It's going to bind to a different type of receptor and there's going to be a different signaling cascade that's activated, all right? So kind of keep that in mind when you're looking at this few slides before this as well. All right, so again, this is an example of another pathway that could be initiated by the binding of a growth factor, okay? And you can see, so this is just giving you an example of a different type of second messenger here, okay? So if we look at this, basically you see the growth factor, right, binding to its receptor. Remember, it's these tyrosine kinase receptors that can autophosphorylate each other. They essentially, once the growth factor comes and binds, the tyrosine kinase forms a dimer, and then again they autophosphorylate, and that sort of initiates this whole process. Okay, so you can again take a look at this pathway here. Nobody needs to memorize, but this is another example of a signaling pathway that would be activated by a growth factor. Okay. And if you notice here, there's a specific sort of protein complex here called mTOR, okay? Now this one is mTOR C2, mTOR complex 2, and you can see it's sort of interacting here with this pathway, okay? And again, when a growth factor binds to the cell, it's basically stimulating proliferation, it's stimulating transcription, it's stimulating translation, right? It's basically causing this cell to become more active and then, like I said, potentially proliferate and make more of itself, okay? All right, so that's that. Now, if we kind of talk a little bit more and show you kind of another pathway here, or right, you can kind of see that some of the players, right, being that AKT and mTOR, are involved in this other pathway as well. Okay, again, we're not memorizing this, but now I'm focusing a little bit more on mTOR, right? So it says the mTOR pathway couples control of protein synthesis. So think of the mTOR as being, if you want a more specific function of the mTOR, is that it's going to stimulate translation, okay? So kind of think of that as being its job, because you can see that in those pathways, there's lots and lots of proteins involved, okay? So it says it couples control of protein synthesis to the availability of growth factors. So when a growth factor binds to the surface of the cell, right here, again, it's going to sort of activate the cell, wants the cell to increase transcription, increase translation, and then maybe, proliferate from there, okay? So you can see here, right, this is showing you that AKT, this is showing you this mTOR C2, like we saw here, right? But then what also is going to be activated is another type of mTOR, okay? And so this mTOR C1 is what is sort of directly, or what directly regulates the translation part of it. Okay, so let's try to think of it that way. So if we want to think about this mTOR C1, think of it as being the protein that's responsible for increasing translation. Okay, and that paper, the paper summary, focuses on this here and tries to understand the mechanism by which some of these things are happening here. Okay, whether and basically it can kind of signal through these two ways, but ultimately this is leading to an increase in translation. Okay, now the paper gets way more involved than we need to be. However, I like to give you papers like that because you know, it just gives you an idea of how specific scientists need to be in order to develop drugs to target a specific protein, right? And how important it is to understand all of the things mTOR C1 might be interacting with, right? And to also understand how this might be different than this here. Okay, so you'll see there is a figure in the paper that sort of looks similar to this and sort of tries to tease out this pathway here. Okay, so I'll sort of leave it at that for the paper, and I'm going to go to the paper in a second just to introduce a few things. But again, remember, I don't expect you to understand everything in that paper. I don't understand everything in that paper. Okay, it's not my field, right? Though I'm a scientist, it doesn't mean scientists are very specific, right? In terms of what they study. All right, so keep that in mind when we're looking at that paper. So just a little bit more here. Again, if we want to think about mTOR's function, it's essentially going to increase translation, right, when a growth factor binds. Okay, so it's sort of the link, and that's what that last slide said. It's the link between growth factor availability and the increase in translation. Okay, so like if this mTOR C1 went away, then maybe you wouldn't see translation being increased. The other effects of some of these things up here in terms of increasing transcription, yeah, maybe that happens, but the translation part of it might not be as robust if this mTOR C1 wasn't around. Okay, so a little bit more on that. The other thing is sort of another function of the mTOR, which coincides with this. So again, what we're showing here is that mTOR C1 will increase translation, okay? When growth factors are around, nutrients, lots of nutrients available, the cell wants to increase transcription, wants to increase translation, and particularly increase translation of proteins that are going to then lead to cell proliferation, right? And cell division, okay? So that's the goal, and kind of that's what a growth factor will trigger, right? And that's why we said that a lot of these pathways and growth factor signaling is involved in cancer, right? In development of cancer. Because when we think about cancer cells, they have increased cell proliferation, right? They need to have the ability to increase translation and transcription in order to keep proliferating, okay? And sort of be overactive. So you can see how these pathways and a lot of these proteins in these pathways are involved in the development of a cancer cell, and how a lot of these proteins would be good targets to treat certain cancers, if you know that this, what's specifically involved. And again, if we think about that paper, that's what it's trying to tease out, a specific interactions and specific responsibilities of these proteins in colorectal cancer, right? In a cancer model. Okay. All right. So the other thing that mTOR will do, and that's what this next slide shows, is that it will also inhibit autophagy. All right. So that makes sense, right? If there's lots of growth factors and nutrients around, the cell's going to increase transcription, increase translation. Well, it makes sense that it would also want to inhibit autophagy. When is autophagy activated? In the exact opposite circumstance, right? Autophagy, remember, is when the cell will break down its own constituents in order to kind of get some nutrients, get some proteins or whatever, because it doesn't have enough nutrients from the outside to increase translation, and doesn't have it can't be active, right? Because it doesn't have the availability of those things is not there, right? So in order to basically recover function and to survive, it needs to break down some of those constituents in the cell that maybe it doesn't need, okay? And so that's sort of a survival kind of mechanism. All right, so if things are good, there's lots of nutrients, there's lots of growth factors around, the cell would inhibit autophagy. And so mTOR is involved in stimulating translation, right? But it also is involved in inhibiting autophagy. Okay, so what this next slide is showing you is now, well, what happens? Okay, so let's go here. If there's lots of growth factor around, lots of nutrients around, mTOR will be stimulating translation. What it also isn't showing here is that it will be also inhibiting autophagy, okay? It's going to inhibit that. However, if we have a different situation now, right, where there is not a lot of growth factors around, there's not a lot of nutrients around, it's showing you low amino acid levels, okay, that will not activate this, right? There's no growth factors binding. None of this is going to be activated, okay? What this is showing you here is that this will inhibit the activity of mTOR, right? So if there's not a lot of mTOR around, so normally, again, mTOR would inhibit this, right? Would normally inhibit autophagy. I told you that, guys, that, right? But say now this mTOR is not around because, again, there's not a lot of growth factors, and so it hasn't, that pathway to basically activate mTOR, it hasn't happened, right? So this isn't happening. So mTOR does not get activated, no active mTOR around, therefore this will be allowed to occur, okay? So the thing, that's why mTOR is such an interesting target, I guess, right? Because it's involved in increasing translation when there's a lot of growth factors and nutrients around, but it also has a dual role in that it controls autophagy too, right? When there's a lot of growth factors around, it will inhibit this, right? So this is when there's a lot of growth factors, right? So don't look at this right now. If there's a lot of growth factors around, right, and basically growth factors are binding to the receptors, and that whole pathway that was on that other slide is activated, there's going to be lots of mTOR around. And what mTOR will do for this process is inhibit it, right? That's why that's showing you this here. So this wouldn't be happening, right? However, on the flip side, not a lot of growth factors around. They're not binding to the receptors. They're not initiating that pathway on the other slide, right? Therefore, there's hardly any active mTOR, right? The low amino acids, no growth factors is basically going to inhibit this, right? This mTOR C1, which then removes this inhibition and allows autophagy to go. Okay? Now, it's confusing. Okay, that's why I wanted to clarify this. And I don't expect you guys to, maybe you're still a little confused. And that's fine, okay? We're not in graduate school. These are super complex. Again, it's meant for you to appreciate how complex these pathways are and the interplay between them, okay? And so anyway, let me just pull up that paper, okay, real quick, and just point out a couple things there, and then that's pretty much it. Hopefully I can find the paper. Yeah, it's right here. Okay, so a couple things, and the other thing too, this is a Nature paper, and so these are very short, which sometimes makes them hard to understand. If you notice, the abstract is sort of seems like the introduction, right? And then you sort of have introduction mixed in with results here. And then there are methods, and then you can see the figures, okay? So again, I don't give you these papers expecting that we understand everything that's going on here. It's about maybe grasping a few concepts from this, but also just understanding how these things are approached in science, okay? So this talks about mTOR C1, right? Talks about mTOR C1's role in increasing translation, right? And tries to tease out how exactly it's doing that, right? And so like I said, let me just do this real quick. Remember I said you kind of have a figure in here that looks similar. Where the heck is that? Right? It's trying to tease out these sort of two different effects that mTOR has, right? And it does this in various different ways in terms of inhibiting certain proteins in those pathways. Okay, so that's where kind of you see there's a whole bunch of figures here where now this is saying, okay, right, this is using a different type of treatment basically to inhibit a different player in that pathway. Okay, I'm trying not to like get into too much detail here. Couple things I just want to point out. APC never really says what it stands for. APC is a tumor suppressor gene, okay? meaning if APC is mutated and not able to function, cells will proliferate more, and that's essentially going to lead to a cancer cell. Okay, so there is certain types of colorectal cancer that have been directly connected to this tumor suppressor gene, specifically this APC. So that if you have colorectal cancer cases and you take a biopsy and you study those cells, you'll see that a lot of times there is a mutation in that APC gene. Okay, so that's what that is, and they're sort of using that as their cancer model. Okay, so in case you didn't know that, again, then they're talking about mTOR activity and its role in translation and how it increases it, and kind of trying to tease out, or talks about kind of whether it's interacting at the initiation part of translation versus elongation. And you can see there's some of these other proteins that we've mentioned in other lectures, or that I've mentioned, okay, and talking about how it interacts with that. All right, but again, big picture, it's APC is the tumor suppressor. If that's mutated in a certain cell line, it's going to mimic or model a colorectal cancer, okay? Then they're looking at now what's mTOR's role sort of in the proliferation part, and specifically in increasing translation. Now, they use various different drugs or chemical agents to inhibit things, right? So for instance, one of the things they use is rapamycin, which will inhibit mTOR, and then look at the effects, okay? So in the assignment, let me just go back real quick here. Hopefully it doesn't pull my paper away. I say describe the main results presented in the paper. I just also added in here, so if you already completed this, you need to just add a little bit. Choose one figure and describe it in detail. Now, there's many panels in each one of these figures. Again, you may not understand every single one, but let's try to look at one figure and try to explain at least one panel from that figure. So for instance, right, you can see, I mean, this paper is crazy, to be honest, in terms of the amount of work that they did and the different specific kind of ways they tried to tease out the mechanism. Okay, I'm aware. Like I said, I don't understand everything in here either. I don't expect you to as well. So, and they also add in this Wnt signaling when they don't really talk about it that much, but don't even worry about that. Yeah, I was going to get into it, but I'm not going to. There was one figure that I just kind of wanted to draw your attention to. And I mean, I guess in terms of something to understand here. So this is basically showing you, right, treatment with rapamycin and then actually looking at tumor regression. And so you can kind of see, so that's probably maybe these two panels are pretty kind of easier to understand. So you might want to pick out one of those. Remember, rapamycin is a inhibitor of mTOR, right? And so that's the idea. Well, if you give rapamycin, you know, as a cancer treatment, we need to make sure that mTOR is involved in the sort of genesis of that cancer, right? And so anyway, this is basically showing you here, if I zoom in on this one panel, oopsie, that's not going to help. Right, if I zoom in on this one panel here, it says % of tumor BRDU positive cells. So BRDU is a proliferation marker. So basically when you give rapamycin, you can see that there's a decrease in the number of proliferating cells here. Okay, this is looking at tumor lysozyme, so that's why you see this increase. Okay, so you can kind of even look at survival, right, rates here. And so anyway, try to pick one figure where you can at least just describe to me the details from one of the panels. Okay, not every single panel in the figure. All right, so again, I could spend an hour discussing this article. We're not going to do it. Again, if we were in class, maybe we would have spent a little bit more time together to go over some of these. But I just want you guys to just kind of appreciate the level of detail that's necessary to understand these pathways. And the level of understanding that's necessary to discover a drug that's going to be an effective cancer treatment, right? And that there are specific pathways in different cancers that might need to be activated, right? So, using a mTOR inhibitor for colorectal cancer may be effective, but for breast cancer, maybe not, right? And so that's why cancers are so hard to treat. All right, guys, I'll leave it at that. So hopefully that helps a little bit. And hopefully by going through those last couple of slides at the end of that cell signaling lecture, help clarify some of that as well. All right,